Every Voice Speaks unites and empowers the muscular dystrophy community by elevating patient voices and fostering collaboration across conditions like LGMD, FSHD, myotonic, Becker, and Duchenne. Through personal stories, expert interviews, and accessible discussions, the show helps listeners understand drug development and learn how their input can influence trials, policies, and care. By centering lived experience, it strengthens advocacy, advances research, and supports better futures for kids and adults living with muscular dystrophy.
Welcome to Every Voice Speaks. Today we're having an important conversation with Sarepta Therapeutics. Last year, our families were devastated to hear that Sarepta had decided to restructure, leaving many of the LGMD programs. Today, we are discussing the future of LGMD R42E gene therapy program that is still on FDA clinical hold. We're discussing what this means for patients and families and where the field goes from here for LGMD R42E. Joining us today are Dr. Louise Rodino-Klapac and Dr. James Richardson from Sarepta Therapeutics. Thank you both for being here and for being willing to engage in this discussion with our community. Before we discuss things with our guests, I would like my co-host Pat Motion to share a bit about his own life with LGMD2E. There are very many subtypes of LGMD, and 2E is ultra rare. Having an audience overview will help us understand what it is at stake in terms of research and clinical trials. Pat?
Thanks, Kat. Yep, thanks, Kat, and thank you very much to our guests, Louise and James. It's great to have you on, and thank you so much for your time today. As Kat said, all the research is exciting. I was diagnosed back in 1985, and as we record this, it's 2026, so that's an awful long time. When I was diagnosed, there was nothing. They did not even know the gene that caused for muscular dystrophy, period. So without drilling down too hard into the science, because that's what our guests are going to do a little bit, let me just say that it is amazing that although it's taken 40 years, we've come very, very far. So all types of muscular dystrophy have specific differences and challenges, and mine is no different. So like Kat was saying, 2E is pretty rare. Some of the limb girdles have heart and lung involvement, some do not. Mine does. So I've been living with cardiomyopathy and pulmonary distress for quite a long time. And I always felt that the disease progression for me was pretty slow. I walked until I was 30 years old. Again, not diagnosed until I was 12. And I did not start having really any heart involvement till probably my early 30s. I started taking heart medication. So I kind of cruised along, didn't start using nighttime ventilation in the form of a CPAP, then a BiPAP until I was in my early 30s. I was cruising along for a long time. At 40, I started using a power chair, but it was still like a very general progression. It kind of felt like somebody walking into the warm end of a swimming pool where I was ready, although disappointed by each slip in progression. I felt I was ready because it was slow. And then in December of 2023, literally without any warning, everything fell off the cliff. It turns out that I was actually having a allergic reaction to a medication I was on, but my heart and lungs had become weak enough that everything went sideways really fast. And I lost consciousness at home for maybe 5 or 6 seconds, woke up, didn't feel right, went to the hospital myself, was there under observation for about 10 hours. They thought I had had a heart attack. They didn't really know, local hospital. Then again, without warning, I went into tachycardia quite severely, and I had to be shocked. And then I was quickly intubated and I was airlifted to Boston, where I woke up and I was intubated on a ventilator, could not speak, could not communicate, did not know really what was happening to me. But while they tried to extubate me a couple of days later, they got all the medication out of my system, tried to stabilize me. I could not deal with being extubated on BiPAP at high pressure, nor could I deal with room air. My lungs had gotten so weak. So the short story of the long story is that I actually coded for 6 minutes, and they thought that they were going to lose me. And my wife thought she was going to lose me, and I did. I had to be resuscitated. I'm happy to say that I'm back to before baseline, but that then they had to give me a trach, and I was still ventilated. I spent 7 weeks in the hospital, 3 at ICU, another almost 4 at rehab. And it really woke me up because I realized that although muscular dystrophy seems to be, in my case, mild progression, I fell off a cliff, and I really thought that was it, and I thought I was going to die. I'm obviously glad that I'm not dead yet, but that was the frightening aspect of what living with my type of muscular dystrophy can do. So, not to put too much of a downer on this, we're gonna drill down a little bit and talk about all the things that the clinical trials are aimed at improving, not only for my type of 2E, but limb-girdle muscular dystrophy in general, and Duchenne and Becker and all the types. We are going to talk about how the rising tide hopefully lifts all boats, and the fact that we're really, again, still thankful and excited that companies are in the space. So that's me. Before we pump it over back to Kat for the question, I would love James and Louise just give us a very brief background of your skill set and how you came to be in the position you're in today. Louise, can we start with you?
Sure. Hi, and thank you for having us here today. So my title at Sarepta is President of R&D and Technical Operations. I've been with Sarepta 8 years, but began my career in academia. So I have a PhD from The Ohio State University in molecular genetics. And then started a postdoc in late 2005 with Dr. Jerry Mindell. So this December, I will be working on limb girdle generally for 21 years. And so my commitment and passion is there for this community. I began my own lab, which really focused and centered around limb-girdle muscular dystrophies. I developed several gene therapies, which we can talk more about. But then when it came time to bring these forward through approval and commercialization, decided to transition to Sarepta, and that's where I still am today, fighting for muscular dystrophies generally, but limb-girdle is part of that. James?
Thanks, Louise. And yeah, thank you so much, Pat and Kat, for having us on today. Uh, Compared to Louise, I'm a relative newcomer to the field. I've been with Sarepta for 7 years, and this is really, these 7 years has been my introduction to the neuromuscular field and neuromuscular research. It's become my passion in those 7 years, and finding treatments for this community has become a mission. But I can't claim um, that before this last 7 years. By older background, I'm a cardiologist and internal medicine physician. Have worked both in clinical medicine in the United Kingdom. Might get my accent's not fully Boston born and bred, and have worked for the last 12 years across different areas of drug development, and that's where my focus at Sarepta is. As Chief Medical Officer, my team are really focused on how we get medicines from the first time they're tested in humans through to providing and producing data sets that let us go and speak to regulators and get these drugs approved and then available to as wide a population as possible.
Thank you, guys.
So thank you so much for that great background, and obviously you guys are serious experts. Louise, can you start maybe giving us an update on Sarepta's LGMD 2E program and the clinical hold and kind of what's going on, and please help families understand what's happening right now.
Sure. So we've been progressing the LGMD2E program for eight years now or more previous to coming to Sarepta. And really the focus over the past several years is conducting a phase 3 clinical trial for the intention of submitting for accelerated approval. Accelerated approval is based on a biomarker. In this case, that's beta-sarcoglycan, which is delivered using a gene therapy vector. And so we completed the first part of the phase 3 trial, which was identifying or meeting the primary endpoint of beta-sarcoglycan expression. And then, because this is a rare population, following the LGMD2E patients in the trial over a longer period of time to then get to confirm that benefit. So unfortunately, in 2025, we had 3 cases of ALF, 2, which is acute liver failure, 2 cases in non-ambulatory Duchenne patients, and 1 patient in our LGMD2E clinical trial, which was a phase 1 trial. Based on that, the FDA placed us on clinical hold because we were using the same gene therapy vector for all of these programs, which is RH74. And until we demonstrated evidence that we could mitigate this acute liver failure, which is preceded by acute liver injury, they placed the program on hold. We were in a position where we were close to submitting the biologics license application, or BLA, as well. We need to come off clinical hold prior to submitting the BLA. But in order to get off clinical hold for LGMD2E, we are conducting a study using sirolimus in non-ambulatory Duchenne patients. And the results of this study are the precursors determined by the FDA in order for us to go back and lift the hold in LGMD2E.
So before we get into that, just
Okay.
for everybody aware, I'm sorry, just for our listening audience, we a lot of people that listen are very, very entrenched in the space, but a lot of people aren't. So I know this is going to be a very layman question,
Sure.
but could you please just explain to our audience, either Louise or James, what's the difference between a phase 1, a phase 2, and a phase 3? So people out there that don't realize how drugs get developed, what does that mean?
Sure. So the question's about the different phases of drug development. So classically, we've, as you said, numbered them 1, 2, and 3. And 1 is the first time you go into humans. And the predominant idea of that is just to test that it's safe. And you will tend to go in at a very low dose, much lower than the dose you expect to end up using in the end. And then you increase the dose slowly, make sure that it's safe. And also, if there are any early suggestions that the drug might be working, that's not the idea of the study, but sometimes you can look at different, normally biological tests that indicate that the drug is doing what you think it should do to affect the disease. Then you go into a bigger study, often one with a control arm, which is called phase 2. And here, it's really is the idea to try to give yourself confidence that the drug works by comparing treated patients to untreated patients. And then if that's successful, phase 3 is a bigger version of phase 2, really, where you are definitively demonstrating the drug works. And also because you're using more patients, you have what's called a bigger safety database. So you have more examples of treated patients and that you help understand the safety profile of the drug better. And it's normally that last study that regulators really look at when they want to decide whether this drug should be approved or not and available commercially, so available to everybody in a given healthcare system. Now, in rare diseases, that's a model that's been around for as long as drug development has. But in rare diseases, there are lots of challenges. In gene therapy, there are challenges. So one challenge in gene therapy is it can be quite hard to test more than one dose. There are, by definition, fewer patients, so it can be very hard to run these bigger studies. And so over the years, working with regulators, those phases have been collapsed down a bit. And so, for example, quite often we'll run that phase 1 and 2 together in one slightly larger study than a phase 1, where we start off by testing safety, and then we try to get that idea, is the drug working? And then we confirm it in another test, in another study. So 1, 2, 3 is the classic way. Often in rare diseases, we squidge some of those together to make allowances for the challenges of having a very small population or a novel kind of treatment like you have with gene therapy.
Right. And I just want to make sure that was outstanding. Thank you. To be clear to everybody, this is why there is a time lapse and a time drag, as people think, why is this taking so long? What's going on? The body needs time, and the trials need time, and we're obviously safety first before everything. And I think people really deserve to know too that the FDA is also being bombarded from anybody and everybody that wants to develop a drug. So it's not as easy as calling the FDA and saying, hey, we have all this data. Can we talk to you tomorrow and present? Like, there's months of planning and all this stuff behind the scenes to even get a meeting. So that's why things take a lot longer than you think that they would. I just want to, because again, I just was at a big meeting of a lot of people that live with muscular dystrophy, and they don't always realize that there are things behind the scenes, that everything has a reason. They don't want to know that. They don't want to stop. They just want to know when the treatment is available. And we have to slow down and tell them there are so many moving parts. It is unbelievable that we've gotten this far. So I know you were going to start talking about the liver stuff, and Kat, please, I throw it back over to you.
Sure. Thank you guys so much. One of the most difficult questions we hear from families is this. Today, ambulatory and non-ambulatory individuals who live with 2E are unable to access gene therapy through this program because of the clinical hold due to acute liver failure or ALF, which I know you want to talk a little bit more about. At the same time, families are seeing gene therapies continuing to move forward in other neuromuscular diseases. And naturally, they wonder why these situations are different. One of the things that they're seeing is ambulatory patients, for example, in Duchenne, are allowed to access gene therapy, even though, of course, it has received approval. But the non ambulatory patients are on clinical hold with the potential of the cohort 8 study. Patients are asking, could ambulatory patients receive gene therapy for 2E if that was approved? Can you help patients understand how regulators evaluate these programs and why different regulatory decisions may occur across diseases and development programs? I think this would be a good question for James, but Louise, if you want to share on this, I think it would be helpful to our communities.
Thank you, Kat. So I guess the question, how do regulators review whether or not to either allow a drug to be dosed in a clinical trial or a drug to be dosed in the commercial setting, I guess, is the key question. And why do some of those decisions seem contradictory to one another? And I think there's an easy answer to this question that's not very satisfying, and there's the more difficult one. So the easy answer is that every drug and data set and patient population needs to be treated and reviewed in its own right. And I think there is a lot of truth to that. And I think that one needs to be cautious when borrowing from very different disease states. I think the more difficult question, which you very specifically raised, is the difference between the 2E population and the Duchenne population, where the similarities are really greater than the differences, right? It's similar disease states and exactly the same viral vector, different gene within that viral vector, but we know that the vector is most likely driving the safety profile. Why do we have this seeming disparity between the fact that the approved gene therapy for Duchenne is still available to ambulatory patients, but we're on clinical hold for ambulant 2E patients? So to think about how regulators view this, essentially what regulators do is that if you'd imagine an old-fashioned weighing scale balanced with two brass plates on either side, they put the risks on one plate and they put the benefits on the other plate. And then if the scales tip down towards benefit, then they will generally allow the drug to be dosed. And if it tips down to risks, they will generally prevent the drug from being dosed, whether that's in a clinical trial or in the commercial setting.
Right.
So if we think about 2E and Duchenne, think about the risks, first of all. We know that the most significant risks are in older, non-ambulant patients. We've seen that in Duchenne. It's probably not really the fact that they're non-ambulant, but actually that they are older, have more advanced disease and more other problems that go with long-term steroid use and the other multi-organ problems you have with Duchenne. We don't know that the risk is zero for ambulant patients. We haven't seen acute liver failure, but we have seen similar levels of liver injury, which is a sort of milder form of that liver failure. So we don't know that the risk is zero in ambulant populations, but we're pretty sure the risk is less in Duchenne, in ambulant patients. I think that it's very appropriate to carry that over to 2E and say that we don't know that the risk is zero. It's probably not zero in 2E ambulant patients, but it is likely significantly less than older non-ambulant patients. So that's the risk side of the scales. And I think it's very fair to borrow those between the two diseases. The other side of the scale, I think, is where the FDA have landed here, and that's on benefit. And this is a little bit more difficult, I think, outside of a regulatory world to understand, because I think that we're all really excited about the benefits that gene therapy can offer to E. Particularly, again, if you compare it to Duchenne, there's actually a biological advantage in this therapy, right? We're replacing the full length gene, whilst in Duchenne, because of the size of the gene and the inability to package it inside the viral vector, we're having to engineer a form of the gene that
Right.
we have a lot of evidence to show that it works, but it's not full length. So why does the FDA view this differently? Well, because really it's how advanced the programs are. So we have the biological theory and we have some biological evidence across both programs, expression. But what we have in Duchenne that we don't yet have in 2E, because we just haven't got there yet, is that we haven't confirmed that benefit in a controlled study where we've looked at how a patient feels, functions, survives. That's the fundamental bar that the FDA sets for traditional approval of medicines,
Okay.
where the biological evidence of expression is interesting, but what really confirms that benefit is that we know that patients perform
Right.
better on certain tasks. We've got some early evidence of that in 2E, but we haven't finished that yet. That's what the phase 3 is for. And until we finish that, the FDA are viewing that benefit side of the scales as lighter than Duchenne. And this is fundamentally a bit of a subjective decision, how you see those scales balance.
Right.
The FDA right now, before we've proven functional benefit, sees the scales in 2E being tilted in the favor of, let's wait to see if you can improve the risk with this new study to look at immunosuppression. Whilst in Duchenne, that additional evidence of benefit we have tilts the scales the other way.
Understood. Thank you. Kat, did you have a follow-up there for Louise?
So thanks, James, for those important comments. I think I want to pivot for just a second, because when we are seeing FDA respond to the LGMDs, it seems as if what's happening in one subtype has affected all the others, which has led to the clinical holds and also the disruption last year that was so devastating. We saw, unfortunately, the death of a patient that was non-ambulatory in the 2D population. And how is that unfortunate death for a non-ambulatory patient affected the 2E program and vice versa? Has it affected going both ways, and has that affected the Duchenne pipeline at all?
All programs, the Duchenne program and all of our limb-girdle programs had used the same RH74 vector. And so during development, it was an advantage because we were able to leverage data from one another. And in this case, we saw the two fatal ALFs in Duchenne, and then the unfortunate fatal ALF in LGMD2D were all in non-ambulatory individuals. And so that's certainly all was part of the decision by FDA to put all of the limb girdles on clinical hold until we were able to sort this out and test the potential to more safely deliver in non-ambulatory patients. And so it all went into their decision. There's a benefit to platform approaches, but in this case also it's in terms of safety, they have to look at everything in terms of the RH74 vector.
Okay. Thank you. Thank you for sharing. And, we understand Sarepta has initiated a new cohort for non-ambulatory Duchenne patients. study in sirolimus immunosuppression regimen in conjunction with Elevidys gene therapy. You mentioned it a little bit earlier, but I think it's important for our community to understand what is sirolimus and how does that mitigate acute liver failure and potentially prevent that. And can you share a little bit more what researchers are hoping to learn and how this could affect the clinical hold with FDA?
Sure. Let me first just talk about the biology and what's happening with the liver and when you deliver this gene therapy. So we're using
Yeah.
RH74, which goes to muscle, which is why we picked it. But whatever doesn't go to muscle then goes to the liver. And this causes, until the capsid is degraded, this causes some local inflammation in the liver, and that leads to the elevation of liver enzymes. Our previous protocol was using steroids, prednisone, to dampen that. But in this case, where we have patients with more advanced disease, um, they're more susceptible to injury. And in the cases of the ALF, we only saw it in patients who are non-ambulatory with more advanced disease. And so we're looking for a way to mitigate this. And so sirolimus, which we're studying in this cohort 8, or the Endeavor study, is what's called a T cell inhibitor. And so this is a way to dampen inflammatory cells. It's been shown in the literature and by others, gene therapy companies, to mitigate the risk of liver enzyme elevation. And so the trial is in approximately 25 non-ambulatory Duchenne patients. The official outcome that we're looking for in that study is a 50% reduction in the incidence of acute liver injury, which is elevation of liver enzymes. And then this is a precursor to acute liver failure. And so that, because Duchenne's a larger population, we could power that study to look for that. And really inform the path forward for LGMD2E and the other limb girdles. And so it's an important study for the Duchenne community, but also important for the limb girdle community as well.
Okay. Pat, I think the next question might be yours. Thanks, Louise.
Yeah, no, and Louise, that's 50% is a very significant number in immunosuppression strategy.
Mhm.
That's great that you're aiming that high. Where do you see the connection between the limb girdle stuff and the broader neuromuscular community and policy work kind of shaping one another? I guess what I'm really asking is, how else do you guys see this connecting the fields where we could progress in one disease, not progress the disease, make progress toward treating the disease in one? I think what Kat and I are nibbling around at is, there's so many different forms of limb girdle that are labeled as such because it's different protein deficiency, whereas Duchenne, we've seen kind of more that you could have Duchenne, but it's kind of spectral, depending on where the deletions are and where the gaps are. But how could one research affect all others? The LGMD community has a lot of self-foundations. So, the 2I people or the 2B people may be listening to this going, okay, that's great, but that's 2E. That's not us. How do you connect all this?
Mhm.
Yeah. I mean, I think the neuromuscular community, the muscular dystrophy community is so interconnected. And when we speak about this connection, we can do it in a couple of ways. So specifically around gene therapy and safety. And in this case, we're using the same exact gene therapy vector. There's just an inevitable connection there that is really important to look at the other indications to be able to help support the path forward for others. I think gene therapy in general, the use of sirolimus and immunosuppression is important across the field. And so whatever we learn in this trial will not only be applicable to our gene therapy programs, but to others, and that might help inform others how they handle it.
Yeah.
And then I
good.
think in the broader sense of limb girdle and the community, when we talk about on the efficacy side and endpoints and how we evaluate trials, how we design trials, I think that is incredibly important to this community. And no one should be looking at an individual subtype or type of muscular dystrophy in isolation. They're all very connected and should leverage one another completely there's just so much learning that can be done. And that's something that we certainly support and will continue to advocate for.
Well, and exactly. And I think what I would want to say too is, learnings in the LGMD space, we certainly would want what we learn in the LGMD space to be able to be translated and help the Duchenne space, um, and other muscular dystrophies as well. So, I kind of am beginning to see where things were very separate, um, just by nature of drug development, was very separate for each individual disease. It's interesting as we continue to understand drug development in the muscular dystrophies, we're starting to see this understanding that what happens with one form of muscular dystrophy may actually affect another form of muscular dystrophy. And that could be a positive, and then that could be a negative. Um, but in general, because the field is advancing in particularly gene therapy, we're also seeing it advance in small molecule therapy. So we're starting to see a whole shift in, I think, the paradigm of how drug development is starting to be perceived. For example, and this is me more as a thought leader in muscular dystrophy than potentially asking a question, but I do see there's this movement amongst the patient community asking these questions of how can we take these timelines down so that we can be more nimble and streamlined so that the cost of developing treatments don't become a factor that creates a scenario where drug developers have to leave this space. And that really leads me to the question that I'd like to ask. It's a difficult but important question. It's probably one you guys really are not excited for me to ask, but many patients and families spent years partnering with Sarepta, and we participated in natural history studies, trials, meetings, community building efforts, because we all believed that Sarepta was committed to LGMD. Looking back, many members of our community felt blindsided by how quickly events unfolded last year. I know that there were lots of factors, but what circumstances led to the decision Sarepta made with regard to the LGMD programs? And can you share a little bit about that, Louise?
I can't say strongly enough how difficult that time was. And it was a series of events that happened in such close proximity to one another. And it was very difficult to navigate through. And I understand how, looking from the outside, how it may have appeared in terms of the cadence of events, and how that we were able to communicate those. We did the best we could as a company. I think one of the things that's important for people to understand is that we were in a predicament for existence as a company. So when the first two ALFs and Duchenne happened, that led to a stopping of non-ambulatory patients having access to the drug, and for a period of time even ambulatory, and that put us
Right.
in a very dire financial position. And so we had to look at the portfolio and stop development in order to continue to exist as a company, because if we didn't exist, we couldn't help anyone. And so that's a really painful reality of where we were at. And so it certainly wasn't trying to divide up or say what's important versus not. It was, how can we exist as a company, continue to drive forward, and find a path forward for the limb girdles, even if we can't be the ones to do it. And so
Sure.
that's really, a year later, obviously I can reflect back on it. It was a very difficult time. And I always put myself in the position of a parent or an individual living with limb girdle and how they feel. And so everyone is allowed to feel the way they're allowed to feel, and everyone has to go through the process. All I can say is, for me personally, for us as Sarepta, we've never stopped driving forward our commitment to limb-girdle. The way in which we ultimately do it may look different, but we are passionate and extremely dedicated to limb-girdle, and we'll continue that. So that's the reality of the situation. I would never want to relive the summer of 2025.
Thank you to both of you for being so vulnerable. And I know from being in the patient community for such a long time, I attend many professional meetings, and you guys are always boots on the ground. There are always members of the team there. Occasionally, reach out for support, and you have reached out for advice of ways to communicate with the patients. And I realize that we have a good working relationship, and that's something that my friends that don't understand rare disease very much are kind of shocked by. They say to me, "Well, what do you mean you can just call up the company and talk to somebody that high level?" And I said, "Well, it's not really like that. It's we're all trying to work on this to help people live their best lives and to help people that have muscular dystrophy." So I don't see it as I'm talking to somebody high level C-suite. I see it as this is the person with the knowledge, and they've made themselves available to us in the patient community. That's another huge seismic change that's gone on over the past 10 or 15 years. I mean, I remember growing up and reading. MDA has a publication called Quest magazine. And when I was a kid, that was really the only way I got my news. There was no internet. So the Quest magazine would come in and I would read about all these researchers and read about all these doctors. And then later, when I grew up going to conferences, I would see these people, and it was kind of like looking someone on a baseball card. And I couldn't believe they were standing in the hallway, as Sarepta does now, where they're just hanging out like, please come up to us, ask us whatever you want. We're going to answer you honestly. That was very shocking to me because I thought all that would be walled off. So in that light, for Louise and James, because we're almost at time for our show, what would you like the patient community, not just in limb-girdle, but in all muscular dystrophies, to know about what's happening currently and where you see the future going? What's coming up? What do you want everybody to know?
I want the limb-girdle community to know that we are still here working very hard for you in multiple capacities. The next milestone is getting data from Endeavor Cohort 8 using sirolimus, and that will help inform the LGMD2E study, allow it to get off hold, and then submit the BLA. We are separately working with the other limb-girdle subtypes to help find path forwards for them, but ultimately the outcome of this next study will help inform all of the limb-girdles. And then in a broader sense, we are here to help support the community. I think it's important to know that we transferred all of our natural history journey data to the GRASP consortium to allow that data to be accessed by other biotechs, other academics and communities to help use that data to support drug development as well. So we are here as a passionate advocate for limb-girdle, both with existing programs and supporting others as well.
I mean, just to add, Pat, harking back to your question, that it's a privilege to be able to meet people like yourself, to meet patients and families that we are developing drugs for, and not just to be working through PDF documents and spreadsheets. That's not
Right.
why anybody does what they do at Sarepta, or I think any rare disease company. This is what motivates us, and I think what continues to motivate us as a company to get these treatments to the limb-girdle community and to the broader neuromuscular community.
Thank you. Thank you, James. And how would researchers access that, or how does that process work for that access to be shared? I guess it's now with GRASP LGMD.
Yes, sure. So it's the data is now physically with GRASP. We're just still in the process of setting up the full administration of that. I think very high level, the data is available free of charge to any researcher who wants to access it. There will be a governance committee, essentially, just to make sure the data is being used appropriately. So GRASP already have this set up for the other natural history data governance, but essentially the researcher will make an application to the steering committee, and assuming that it is a scientifically valid use of the data will be provided to the researcher.
Okay, that's great to hear. And then as we just get to our last question, What do you feel that the FDA needs to learn in order to resume progress for non-ambulatory Duchenne patients and non-ambulatory LGMD or even ambulatory LGMD 2E patients? Where do we think that? And in terms of innovation, it's like kind of two-part question. We've been advocating for basket trials, external controls, platform approaches. What innovations do you believe have the greatest potential to accelerate therapies for the whole field?
So in terms of what the FDA need to learn, as we need to learn as a community, we just need to understand what more we can learn about this risk of acute liver failure, both in terms of increasing our understanding of why it happens and how we can prevent it. And though the sort of chief thing amongst that is this cohort 8 study from the Endeavor cohort 8 and the Endeavor study in Duchenne, that's going to be very helpful in adding to the evidence base that we can help prevent ALI, acute liver injury, and acute liver failure through increased immunosuppression. That's one, I think, in the sense of a body of evidence that we and the rest of the community are creating to better understand this problem and understand how we can better, what we call, mitigate it, so reduce the risks of it happening. And so we need to put all that evidence together and have a conversation with the FDA and say, look, we think that we can, what we call, manage this risk. We can look after patients or provide guidance to patients that will reduce this risk. That'll really help, I think, with that risk side of the scales I was talking about before. And then any evidence, whether it's in 2E or in Duchenne, also to look at the benefit side of the scales and can we provide more evidence to show how this drug is helping patients. And overall, we hope to tilt the scales to a place where the FDA feel comfortable in providing this medicine, whether it's in 2E or these medicines, whether it's in 2E or in Duchenne. And I think the FDA have been collaborative throughout. It was a tricky period for all of us last summer, but we've had nothing but a collaborative and scientific relationship with the review group there. And so I'm confident that if the data supports what we're talking about, which I think it will, then we'll be able to have that conversation and be successful with them. The question about what we need to think about in terms of innovation, I can, from a regulatory and a development hat, two things. Like, I think regulators need to continue to be flexible. What does that mean? I mean, it means be a little bit less old-fashioned and a little less wedded to the traditional ways of developing drugs and accept different kinds of evidence, accept an argument that's drawn from lots of different pieces of evidence, which we call totality, rather than one individual trial. I think continue to innovate in the technologies that we use. I think MRI has been fantastic. I think AI will be very helpful. I know you can't have a podcast now without mentioning AI, but it lets you look at more data in a more objective fashion than you could do with traditional measures. And that can be really helpful when you're limited with the number of patients you get into a trial. So if you can't get more patients in a trial because the disease is ultra rare, maybe you can get more data out of that trial by either using technologies like MRI and using technologies like AI that helps you look at more data points and pull them together and make sense of them. So I think that's what I'm most excited about. I think that's what will most help the rare disease population in general.
Thank you for that, James. Thank you very much for coming on today. Thanks, Kat.
Thanks, Pat.